Hi everybody, nice to see you. Hi, you're on time. Nice to see you. If you can hear me, put a plus in the chat, just so I know that you can hear me and everything's working fine. That would be great. Just put a plus in the chat for me. Hi everybody. Okay, so this is our last A-level biology free stream. We're going to be doing the nitrogen cycle today, so thanks for joining us. Yeah, I'll give you a few seconds. Just put a plus in the chat if you can hear me. Hi, Darcy. Nice to see you. Hi. Okay. Right, so the aim of our lesson today is to go through the nitrogen cycle and hopefully just clarify all of the different stages. So by the end of the stream, you'll be able to draw your nitrogen cycle, describe all of the stages and all of the different bacteria that are involved. Hi Celeste, nice to see you. Okay, so if you're doing AQA A-level biology, then you cover both the nitrogen cycle and the phosphorus cycle. If you're doing OCR A-level biology, you'll know that you do the nitrogen cycle and the carbon cycle as well. So we're going to go through the nitrogen cycle today, which is one of the important nutrient cycles. At the end of the presentation, I'm going to talk to you a little bit about our January A-level biology course, which if you've been here before, you'll know it's our A-level exam preparation course that we're launching in January to help you prepare for your A-level exams. Okay, so if you could just type something into the chat, let me know a little bit about yourself, whereabouts in the country you are from, what subjects you are studying, what bits of biology you find most difficult, anything like that, just to kind of introduce yourself. Let me know that you're here. And I will tell you a little bit about myself for anyone that is new to our biology streams. So my name is Laura. I am a biology teacher. I've been teaching for over 15 years. I've taught in both schools and in sixth form colleges. I obviously did A-level biology and then went on to study biology at university. I went to Manchester University. From there, I went straight into teaching. So I did my teaching qualification in secondary science. And since then, I've been teaching students right the way through from year 7 to year 13. Most recently, I've been head of biology in a sixth form college. So I've been focusing on A-level biology, both AQA and OCR. And I've also done a lot of work with biology exam boards. I've done a lot of marking for the exam boards. So hopefully I will be able to give you some insight into how to best answer the questions and how to make sure you get maximum marks so that all of you can get A and A-star grades in your A-level exams. That's the aim, isn't it? We all want to get those top grades so we can go on to our chosen university courses, which I know a lot of you will be wanting to do. Science, hopefully biology, because it is the best science subject. Okay. So the rules to follow, we don't have many rules for these streams, but the most important is that you you ask all the questions that you want to ask. You can type them into the chat. I will be watching the chat throughout the stream. So if you've got any questions, I will answer them for you live in the lesson. Or the moderators that we've got in the chat as well will help me if I miss any of your questions, or if you've got specific questions about the course that's launching in January, The moderators will be able to answer those questions for you. So feel free to engage, type as much as you like into the chat. And there's no silly questions, that's what we always say. So just ask away. This is your opportunity to make sure you fully understand this topic when maybe you didn't fully understand it the first time you went through it in school. We've also prepared some study notes for you. I don't know if any of you have received your study notes from previous lessons. Let me know if you have in the chat. Let me know if they've been useful. But we've prepared some study notes on the nitrogen cycle, and they pretty much match up to our presentation today. So you can go back through it and obviously have a go at answering all the questions in there and doing some extra revision after the live stream. Okay, let's get started. We've got quite a complex topic to go through tonight, but as always, we're going to simplify it down and make it really easy to understand so that you all feel confident after the end of the lesson. Right, first thing we need to consider then is, why is nitrogen so important? Why are we bothering to learn about the nitrogen cycle? Well, it's because all living things need nitrogen. All living things need nitrogen to make certain compounds that contain nitrogen. So I've just given you a few examples here of biological molecules that all living things need to make. And to make these compounds, we would need nitrogen. So you'll already know, if you're in year 13, that amino acids, which make up proteins, contain nitrogen. DNA obviously contains nitrogen, as does RNA. Can anyone tell me which bit of the DNA nucleotide or which bit of the RNA nucleotide contains nitrogen? Put your answers into the chat. Which part of those nucleotides actually contain the nitrogen? Let me know if you can answer that. We've got some other nitrogen containing compounds as well, just hidden behind my chair. We've got ATP. ATP contains nitrogen. Again, let me know if you can tell me which part of ATP contains nitrogen. And I've put a fifth example, which is NAD. I've included that example because if you were here a couple of weeks ago, we talked about aerobic respiration and we talked about a coenzyme called NAD. That also contains nitrogen. So that is something you could list as a nitrogen containing compound. Do remember to write in the chat. I can't see many answers currently, but I'm sure there are lots of you there that know which bit of DNA, which bit of RNA contain nitrogen. Let me just give you a little bit of help here. So DNA, just find that. Okay, so DNA is made of nucleotides, right? Same as RNA. RNA is also made of nucleotides, but which bit of the nucleotide contains nitrogen? It's this bit here, isn't it? This bit here is the base. Yeah, good, thank you. This is the base, and we've got an RNA nucleotide over here, that's the base. And you'll remember, in fact, Shrey's just said it, we call it a nitrogenous base because that's the bit that contains nitrogen. Okay, so we've got nitrogen in DNA in the base, we've got nitrogen in RNA in the base. We all know the amino acid, right, contains nitrogen. Do a bit of revision of that while we're here as well. Here's my amino acid. And look, that contains nitrogen. So all of these things contain nitrogen, which is why the nitrogen cycle is important, because all living things need nitrogen. You could be asked in your exam to name 2 compounds that contain nitrogen. So you'd have to give 2 of these 5 possible answers. Okay. Right, so 78% of our atmosphere is made of nitrogen. So you'd think it would be quite easy for us to get enough nitrogen, but it's actually not, because plants and animals can't make use of nitrogen gas. What has to happen is the nitrogen gas, which is N2, so that's nitrogen gas in the atmosphere. Nitrogen gas first has to get into the soil so that plants can actually absorb it. And then obviously animals eat the plants, and that's how we get our nitrogen. Or we eat other animals, that's how we get our nitrogen. But 78% of the atmosphere is nitrogen gas. But like I say, we do need to get it into the soil before living things can actually make use of it. So we're going to go through how that works. And we're going to look at the 5 main stages of the nitrogen cycle. So I've given you the names of the stages here. Now, currently this will seem like gobbledygook. It looks very confusing, very complicated, the words, but we're going to look at a diagram which is going to help us understand what these 5 main stages actually are. Okay. So this diagram, I think, is the best diagram of the nitrogen cycle. You'll see lots of different diagrams, and if you look in textbooks and revision guides or online, can be quite confusing. But I always like to use this diagram because I think it's simple, it's easy to follow. So hopefully this will help you, especially if you get the study notes. You can learn this one off by heart. Let me know in the chat if you have already learned the nitrogen cycle. So if you're in year 13, just let me know if you've already covered this in school. And if you have, maybe let me know how you found it. Did you find it difficult? Did you find it quite easy to understand, or was this something that you struggled with? Let me know. I'm sorry about my voice today, guys. I'm absolutely full of cold. I don't know if any of you are. Just in time for Christmas, I've got a runny nose and a sore throat, so I sound a little bit hoarse. Hopefully we'll be better by Christmas. Celeste, how did you find it, Celeste? Did you find it straightforward? Did you struggle with it? Hi Joel, how are you doing? Oh, Joel is one of my students. Nice to have you here, Joel. Joel, I've already taught you this, so you should be an absolute expert. You should know this off by heart. It depends what exam board you're doing. What chapter is this a part of? If you're doing AQA, it's module 5. So let me know what exam board you're doing. Right. Let's make a start then. We're going to go through, we're going to split it into 5 stages because you need to chunk it up. It's too big a topic. If you try and learn it all in one go, you'll probably overwhelm yourself. So what I say to my students is chunk it up and memorize it in chunks and then piece it all together. Celeste saying not too bad. Good, that's good to hear, Celeste. Well, hopefully after our lesson today, you'll be a real expert in it. Right, let's start with nitrogen fixation. So nitrogen fixation is this stage here, okay? So basically, with nitrogen fixation, we're going to take nitrogen gas. Remember, there's 78% of it in the atmosphere, but plants and animals can't use it. So we're going to take that nitrogen gas and we're going to convert it into ammonia in the soil. So let's have a look at how that works. I'm going to put the next slide on. Before you look at the text, I know there's a lot of text there, so before you look at that, let's just simplify it over here. We're going to take nitrogen gas in the atmosphere and we're going to convert it into ammonia. Now, if you're a chemist and you like using formula, ammonia is NH3. See, I'm a biologist, so I tend to use words, and that is fine. I think I'd be worried in the exam that I would forget all the chemical formula. So I like sticking with words. I think us biologists do that a lot, don't we? So you can just say nitrogen is converted to ammonia. That's absolutely fine. But if you want to say N2 is converted to NH3, you can do that as well. Hi Shakira, nice to have you here. Okay, so that's nitrogen fixation. So ignore all the words for now, because if you can just say this, if you can just say nitrogen gas is converted into ammonia, that's the gist of it. It's that simple. The reason we've got some more text over here is because there are two different types of bacteria that do this, okay? So they're all called nitrogen fixing bacteria, because don't forget the stage is called nitrogen fixation. So we call them nitrogen fixing bacteria. There are two different types, okay? So some of them are just in the soil. We call them the free living nitrogen fixing bacteria, and they are just in the soil, and in the soil they convert nitrogen gas into ammonia. Others, our second type of nitrogen fixing bacteria, actually live inside the root nodules of certain plants. Not all plants have nitrogen fixing bacteria in their roots. It's just a group of plants called the legumes, or the leguminous plants. Leguminous plants are things like, I've put peas, beans and clover, and soya plants as well. So they actually have these bacteria inside their roots, which does give them a big advantage, because they can convert nitrogen gas into ammonia inside their own roots. So they're not competing with other plants for ammonia in the soil, because they've got their own little army of bacteria in their roots that can make ammonia for them from nitrogen gas. I'm just going to show you a picture actually on the next slide. We'll come back to that in a second. But this is what I mean. So these are obviously roots of a leguminous, can't spell that, leguminous, there we go, leguminous plants. So plants like peas or beans or clover that are what we call legumes. And can you see these root nodules here? So these are root nodules, and in the root nodules is nitrogen fixing bacteria, okay? And remember what that nitrogen fixing bacteria does, just repeat it over here, converts nitrogen gas into ammonia, okay? So that's stage one, okay? Let's just go back for a second. Okay, so this is stage one. This is what we've just covered. Nitrogen gas in the atmosphere is converted into ammonia, or NH3. The only extra thing to remember is there are two types of bacteria that can do this. We've got the free living nitrogen fixing bacteria, and we've also got nitrogen fixing bacteria in root nodules of legumes or leguminous plants. Okay. Has anyone heard of the word mutualistic? As if I just go back to this slide, these nitrogen fixing bacteria are what we say, we call them mutualistic. Does anyone know what that means? Do you want to type it into the chat and let me know? We say that these ones are mutualistic. What does that mean? Mutualistic. Just think about the word mutual. That's going to give you a clue as to what it means. Celeste, you've done this before. Joel, I know you've done this before. What does mutualistic mean? Okay, because they're in here, the bacteria, they're in the root nodules. They are fixing nitrogen gas into ammonia, but we say that they have a mutualistic relationship with the plant. I'm not sure anyone is able to answer this question. We're not sure what mutualistic means. It basically means that both the species benefit. They're both getting something out of the relationship. It's a mutualistic relationship. Oh, okay, Celeste, yeah. So mutualistic, like if something's mutual, it's like, we use it to mean like they're in agreement, but in this sense, we're saying that they both get something from the relationship. So the plant is obviously gaining because the plant is getting a source of ammonia, but the bacteria are gaining as well because the bacteria actually can get glucose from the plants. So as you all know, I'm sure, plants make glucose in respiration, and the bacteria can actually use that glucose for their own respiration. So the plant is getting a source of nitrogen from the bacteria, and the bacteria is getting a source of glucose from the plant. So if you see that word in future, mutualistic means that they're both kind of benefiting from the relationship and living kind of in harmony, together. Let's say it like that. Okay. Right, so that's nitrogen fixation. I'm going to just build up, actually. I'm going to build up my nitrogen cycle over on this side because it's always handy to have a little sketch. Have a little sketch of it as you're learning about it. Okay, so we've done stage 1, which is nitrogen fixation. Now we're going to do stage 2, which is nitrification. Okay. So nitrification is a 2-step process. So we've got nitrogen gas, we've fixed it into ammonia. With nitrification, we're going to take that ammonia and we're going to convert it into nitrites. Again, for the chemists, I suppose you might want the chemical formula. You don't have to use them if you're not confident with them as biologists. You don't have to use them. But we're going to take ammonia, which is our NH3 here, and we're going to convert it in 2 stages. So it gets converted into nitrites and then into nitrates, and this is called nitrification. So it makes sense that it's done by nitrifying bacteria. Okay, so it's called nitrification and it's done by nitrifying bacteria. It's actually an example of oxidation. Hopefully you can see that once you look at the chemical formula. So you're gaining oxygen here and then you're gaining another oxygen here. So it's an example of oxidation. So it occurs well in aerated soils. Basically soils that have a lot of oxygen in them, you'll get a lot of nitrification and a lot of nitrifying bacteria. Has anyone ever seen, farmers kind of ploughing the fields, turning over the soil? That is helping to aerate the soil or get oxygen into the soil because it does help with the fertility of the soil. If it's oxygenated, then this process of nitrification will happen more and you'll end up with more nitrates in the soil, which is what we want for plants. Okay, I'm just going to go back to our cycle. So we've done stage 1, nitrogen fixation by nitrogen fixing bacteria converts nitrogen gas into ammonia. We've now done stage 2, which is this one here. Ammonia is converted into nitrites and then into nitrates. And you do have to know it's a two-step process, and that is done by our nitrifying bacteria. And it is an example of oxidation. Can you tell me, actually, I'm just thinking about different exam boards. Can you let me know if you do OCR biology? Is there anyone listening that does OCR? Because if you do OCR, you do have to know the specific names of the bacteria as well. So let me know, because if there's anyone in the chat that does do OCR, I can give you the specific names. For AQA, we can just call them nitrogen fixing bacteria and nitrifying bacteria. But if there's anyone that does OCR, I can give you those specific names. So let me know. Okay. Right, so stage 3. Stage 3 is the easy stage. Does anyone know what this word means, by the way? Assimilation. If we assimilate, what is assimilation? Just in general, in the English language, what does it mean to assimilate something? Let me know what assimilate means. Oh dear, you do OCR. Okay, well what we'll do at the end, Dia, is we'll go through and we'll add in the specific names of the bacteria, because unfortunately for you, you've got to know it in a little bit more detail. Okay, so what do we mean by assimilate? Absorption. It follows absorption, Celeste. So plants, once we've got the nitrates, plants will absorb them and then they will assimilate them. So it follows on from absorption. So once we've got the nitrates, in fact, Celeste, I'll put absorption there as well. Once we've got nitrates, they are absorbed, usually by active transport, because it's usually against the concentration gradient. Yeah, but then after they're absorbed, we say they are assimilated. Okay, so the key to assimilation is this. When you assimilate something, you're basically making something, you're building something up. So in this stage, the plants are basically using the nitrates that they do absorb. You're not wrong. They absorb them and then they use them to build or to assimilate things like proteins, their DNA, their RNA, their ATP, all of those compounds that we spoke about at the start of the lesson. They can use those nitrates as the source of nitrogen to build all of those important nitrogen containing compounds. Okay, let's move on. Now, before we go any further, I'm going to do the little sketch again. So follow with me and check that you've learned this so far. Okay, so we've got, this is what we've done so far. I'll put both, I'll put absorption and assimilation. Okay, so we've got over here, we've got stage 1, which was nitrogen fixation. Over here, we've got stage 2, which was nitrification. And over here, we've got stage 3, which is absorption and assimilation. Shakira, yeah, assimilation refers to the incorporation. Yeah, so they're incorporating the nitrogen into their nitrogen containing compounds. And once they've absorbed and assimilated, they can make protein out of that, you know, amino acids, they can make DNA, RNA, ATP, etc. Right, we're now on to stage 4, which is ammonification. So plants, unfortunately, all plants are going to die. as are animals. Should I put some animals on here as well? That's obviously just feeding, that stage there. So plants get eaten by animals. Now, unfortunately, all animals and all plants will die, and that will add more ammonia back into the soil, which is what stage 4 is all about. So the protein in dead organisms, okay, so protein in dead organisms gets converted into ammonia. Now that's done by some bacteria that are called the saprobionts. Does anyone know what a saprobiont is? Let me know in the chat, what is a saprobiont, or what is another word that you may have used for a saprobiont? Sorry, just full of cold. Let me know what a saprobiont is, if you've heard that word before, or maybe your teacher used a different word. They're a group of bacteria, but do you know what type of bacteria they are? Let me know what you think. Yeah, exactly. Celeste, they are decomposers, which basically means they are the bacteria that cause decay or decomposition. They break down dead plants and dead animals. So what they're doing here are saprobionts, or are decomposers. They are breaking down dead plants and animals, and they're breaking down the protein that's in them, and they are converting it into ammonia. I've got a slide here actually that just tells you a little bit more about saprobionts. So they are microorganisms, which you'll know means microscopic living things, also known as decomposers, and they basically decay organic matter. So plants and animals that were once living, that contain a lot of carbon, that's what organic means, they break down dead plants and animals. They actually do this by secreting enzymes. They use extracellular enzymes. What does extracellular mean? If the enzymes are extracellular, what does that mean? Whilst you're answering that, I'm just going to summarize this stage at the top. What does extracellular mean? No answers yet. I'll give you a little bit longer because I'm sure you all know what extracellular means. More than one? It's not more than one, although there is more than one. There's definitely more than one enzyme involved, but that's not what extracellular means. Let me just do a quick. Out of the cell, yeah, that's exactly what it means. Out of the cell. They secrete them outside of the cell. Extracellular. That prefix extra always means like outside of, doesn't it? Like extraterrestrial means like outside of Earth. Extracellular, outside of the cell. I'm just going to show you a little bit more about how this actually works. So they secrete extracellular enzymes, these saprobionts. Firstly, they secrete protease enzymes. And protease enzymes will obviously digest the protein that's in the dead plant or animal into amino acids, which you should know the structure of. And then the second enzyme breaks down the amino acid, and look, that's how we get ammonia. Can you see that? So the second enzyme they secrete extracellularly breaks down the amino acid, It removes the NH2 group and this hydrogen, and that is ammonia. Okay, now you don't need to know it in that much detail, but Shakira, you were right. There is more than one enzyme. You've got the protease enzymes that break down the protein into amino acids, and then enzymes called deaminase enzymes break down the amino acids into ammonia. But we can simply summarize that just by saying saprobionts break down protein in dead plants or animals into ammonia. So that's a summary of that stage, which is called ammonification. Okay, last stage before we try and put it all together. I know we've done an awful lot, but we will go through it as one cycle in a second. We've got the final stage, which is denitrification. And this is the stage that we don't really want to happen, because denitrification, which is done by denitrifying bacteria, these bacteria convert nitrates in the soil back into nitrogen gas. Now, this is going to be a disadvantage for our plants, because once we convert nitrates in the soil back into nitrogen gas, the plants cannot absorb and assimilate that anymore. So it's basically a loss of nitrogen from the soil. It represents a loss of nitrogen, and the soil will decrease in fertility, less fertile, because it's got fewer minerals in there now. These bacteria are actually anaerobic. They use anaerobic respiration, so they thrive in soils that have little oxygen. So if a soil is waterlogged, so full of water, which has got a lower concentration of oxygen in it than air, these bacteria will thrive, which is a bad thing for the plants, because the nitrates will be lost to the atmosphere. They'll go back into nitrogen gas. Okay, let's start putting this together. So we need to learn the names of the stages and the bacteria that carry out that stage. Now, obviously there is a fifth stage, which is assimilation, but I've not included that on the table because that does not use a bacteria. That's just when plants absorb and build things with the nitrogen. So that does not involve a bacteria. But in terms of the stages that need bacteria, we had nitrogen fixation, and we call them nitrogen fixing bacteria. Nitrification, which is our nitrifying bacteria. Ammonification, which was our saprobionts that break down the protein in dead plants and animals. And denitrification, which is our denitrifying bacteria. Now, someone before said they do OCR, which unfortunately, I wasn't going to go through this, but seeing as we've got someone, there was at least one of you that does OCR biology. If you do OCR, you do need to know the specific names of the bacteria. So I'm going to give you some examples. Our nitrogen fixing bacteria, Azotobacter, are the free living ones that we said were in the soil. Rhizobium are the mutualistic ones that are in the root nodules. Oh, someone else does OCR. Nitrifying, you've got Nitrosomonas that convert ammonia into nitrite, and Nitrobacter, which convert nitrite into nitrate. Decomposers, we can all just call them decomposers or saprobionts. Denitrifying bacteria, the example that we get given for OCR is Pseudomonas. I mean, there's loads of denitrifying bacteria, but one of the main denitrifying bacteria is the Pseudomonas. Dear, these aren't on the study, well, the slide is on the study notes. My written notes aren't on the study notes. When I wrote those notes, I was thinking mainly about AQA biology. So if you want to jot the names down now, you can do that, because you do have to know specifically which bacteria are which. So there are free living, nitrogen fixing. There are mutualistic ones. Nitrosomonas convert ammonia into nitrites, and Nitrobacter convert ammonia into nitrates. So there's a little bit more to learn, unfortunately, for our OCR biologists. There's a little bit more content. I always find with OCR biology there is more content, but you don't have an essay. That's the payoff, isn't it? Those of you that do an AQA, I'm sure you've already started practicing, but you have to write an essay at the end of paper 3. OCR biologists don't, but they do have a little bit more content to learn. Right, so here's our summary. Can we name the main stages? I've numbered them 1 to 5. Can you put into the chat the names of those stages? Can you remember the name of each? Put your answers into the chat, and then we'll use this diagram to just kind of put everything that we've learned on there. I'm going to have a drink before I totally lose my voice, I think. How can you access the study notes? If you stay on till the end of the lesson, you should be able to access them. Just if you ask in the chat, one of the moderators will let you know at the end how you can get these study notes. Okay, so put your answers into the chat. I want you to put in the name of stages 1 all the way through to 5, which are the 5 main stages of the nitrogen cycle. Whilst you're doing that, let's just add in some more information that we've covered today. Okay, so I've given you all the extra information that you need for that stage. Let's put a bit more on for this one as well. Thank you, Celeste. Oops. Celeste's got all the stages in there. Okay, so this is the kind of thing you should do with your revision. You do a sketch of the nitrogen cycle, you make sure you've got all the key processes, and then you go through and you annotate it with all the extra bits and bobs that you need. Okay, so Celeste has given us all of the answers. Well done, Celeste. So stage 1, we've got nitrogen fixation. Bit of a clue, because the bacteria are called nitrogen fixing bacteria. So that's nitrogen fixation, and it's nitrogen gas into ammonia. Just remember there are 2 types of nitrogen fixing bacteria, free living in the soil or in the root nodules of leguminous plants. Stage 2, we've got nitrification. I think you've got them all right, Celeste. Yeah, I think you've got a couple in a different order, but it doesn't matter. I mean, it is a cycle, so you can kind of describe it, and you can start anywhere with the cycle, can't you? Stage 2 is nitrification. Ammonia into nitrite, nitrite into nitrate. Remember that is oxidation, so that happens in soils that have a lot of oxygen, aerated soils. Stage 3 is where the plants absorb the nitrates, and it's called assimilation, because once they absorb the nitrates, they can assimilate them, so they can use it to build proteins, for example. Animals obviously eat the plants, so we've got consuming on there as well. Stage 4, yeah, Celeste, you've got that, ammonification. So stage 4 is ammonification. This is done by our saprobionts. Remember, they use extracellular digestion. So they are converting protein, should I put dead there? Dead plants, dead animals. They break down that protein and they convert it into ammonia. And then look, we're back here. So then that ammonia can undergo nitrification again. So you've almost got like a cycle within a cycle going on here. Stage 5, we've got denitrification, which is the stage that plants don't really want, because this uses anaerobic bacteria and they convert nitrates back into nitrogen gas, which means the plants can no longer use it. So it's a loss of useful nitrogen from the soil. Okay, so that is our cycle. Let's have a go at these questions. So we've got some straight recall questions first, and then we're going to look at a couple of harder questions. Put your answers into the chat. Question 1, one reason why plants and animals need nitrogen. And then table question number 2, I've given you the name of the process, but can you name the bacteria responsible? Just the general group name of the bacteria. I can go through and give the specific names for our OCR students as well. But for now, let's just do the kind of group name for the bacteria. So put your answers into the chat. Hopefully question 1 is okay. Should be able to name like one compound at least that plants and animals would need nitrogen to make. Have a go. I think I gave you about 4 or 5 different compounds that plants and animals need nitrogen for. So you should definitely be able to give one of those compounds. And then think about the bacteria as well. Put your answers into the chat. I know Celeste, you're going to answer it for me. Dia can have a go at answering it. Who else have we got? Shakira. Joel, I don't know where Joel's gone. Yeah, good, we're getting some answers. So yeah, they use it to make proteins. There's still more that we can say though, guys. You can give me some more answers. What else have you said? DNA. Tommy, they don't need it to make ammonia because that's not a compound that they need. But there are some others. For example, we had RNA. We also had ATP. We also had NAD, which is a coenzyme that they need for respiration. For plants, you could also have NADP. That's a coenzyme that plants need for photosynthesis. So that's plants only, that one. So you couldn't have that for animals and plants because that one's plants only. Thanks, Joy. Right, let's go with Joy's answers. Oh, I think Joy's answered the previous slide. She's given me the names of the stages. And here we're looking for the names of the bacteria. So, but I mean, the stage names give you a clue. These are our nitrogen fixing bacteria. Celeste, yeah, these are our nitrifying. Oh, I've done that in the wrong place, guys. Confused myself there because they're in a different order. So these are my nitrifying bacteria. Let's see if I can rub that out. Yeah, so they're my nitrifying bacteria for nitrification. Ammonification. Yeah, Celeste's on the ball as always. Saprobionts. Denitrification, yeah, denitrifying. D-night, I've spelled that wrong. What is going on? You can tell I'm not well today. I'm losing it. Denitrifying. Denitrifying. Sometimes it's hard to talk and spell correctly at the same time. Denitrifying bacteria. Remember, if you're OCR, you do need the specific names. Is anyone here still from OCR? Or did you switch off when I said you had to learn the names? It's a lot, isn't it? It's a lot. Rhizobium. Unfortunately, guys, you've got to learn these names. I'll just put a circle around those because they go with nitrification. Okay, so we've got nitrogen fixing bacteria. Azotobacter are free living, Rhizobium are in the root nodules, and then for nitrification you've got Nitrosomonas followed by Nitrobacter. For denitrification, AQA can just say denitrifying bacteria, but OCR are going to call it Pseudomonas. Okay, last couple of questions then. So these are more exam style questions. You might get a 5-mark question that literally just says describe the nitrogen cycle or describe the cycling of nitrogen. And if that's the case, you just go through it exactly how we've learned it today. You can use bullet points, you can use numbered steps, you go through the whole cycle, naming the stages, naming the bacteria, whole cycle. But quite often what they do is they'll give you shorter answer questions where you just have to pick the right stage. So you've just got to think, right, which bit are they asking me about? And then you zoom in on that bit. So I've zoomed in, if you like, on nitrification. So describe the process of nitrification. 2 marks. Write your answers into the chat. And then with the next question, I've tried to get you to focus on a different stage. Describe and explain what would happen to the nitrate concentration in waterlogged soil. So waterlogged soil, have a think about which stage I might be getting at there. Which stage of the cycle uses anaerobic bacteria that would thrive in waterlogged soil with low concentrations of oxygen. Have a little go at those 2 questions. Put your answers into the chat. Meanwhile, just to help you, I'm going to do a little quick summary here. And this is the stage that you should get to. You've got to be this confident. So someone can give you a piece of paper and you can say, yeah, here's the nitrogen cycle. Okay, you've got to sketch it like this. And obviously label the stages and name the bacteria, but you should be able to get it down like that. Right, let's have a look at the answers. So describe the process of nitrification. Nitrifying bacteria turn ammonium ions into nitrites, into nitrates, and this is oxidation. Perfect answer. Perfect answer. It's this bit here, isn't it? We are zooming in on nitrification, which, as Celeste said, is ammonia or ammonium ions, you can say either, into nitrites, into nitrates, and that is oxidation. Well done. And it uses nitrifying bacteria. Yeah, that would be my tip actually. Every question, you should name the stage, name the bacteria responsible, and then give an overview of that stage. Now, if you look at my question here, I've already named the stage, so that's not going to get you a mark. But for 2 marks, you should be able to name the bacteria, so nitrifying bacteria, and give an overview of the stage. So ammonia to nitrites to nitrates. Let's have a look at the other answers for question 4. Waterlogged soil denitrifying bacteria in the soil break down nitrates and return nitrogen gas back to the air. Perfect. Denitrification, where denitrifying bacteria thrive in anaerobic conditions, so convert nitrate ions back to nitrogen gas in the air. Perfect. We're looking at this stage here. So this stage, so you're going to get a mark for naming the stage, denitrification. It's done by denitrifying bacteria because they thrive in waterlogged soil because they are anaerobic. And then give an overview of the stage, as you did perfectly. So they are converting nitrates, or NO3 minus, back into nitrogen gas. So it's bad for the soil, it's bad for plants because there's going to be less nitrates left in the soil. Okay, well done. I know that's been a tough one tonight, but I'm hoping after the lesson you're going to get a piece of paper, you're going to sketch the nitrogen cycle, and you're going to write everything on there. The names of the stages, the names of the bacterium, and any additional information that you can remember. Well done for all your answers. Let me talk to you about our course that we are launching in January. Some of you may already know it's for students that want to get the best grades possible in their A-level biology. So if you're aiming for that A or A star and you want a little bit of help with revision, I mean, it's more than revision. We're going to go over all of the content. So we're going to go over all of the content again, because sometimes it is just not enough to go through it once in school with your teacher, particularly for biology, because there is so much content. So we're going to go through all of the content again. We're going to go through lots of past paper questions. The course will basically include 2 live lessons every week, which will take place on YouTube, and each lesson is going to run for 2 hours. So we'll get time to go through the content and do more questions. In these lessons, in these free streams, we don't get time to do many questions, but in our course in January, we'll do more past paper questions. Okay. You can also get access to 24/7 mentor support. So our mentors are amazing. They've all got A's or A stars on their A-level biology, and they will be there to help you whenever you need them. Any biology related questions, homework support, revision tips, guidance, anything like that. They will be available to you directly through chat. So that's a really, really great support, and it's really unique to this course. You will get exam style homeworks. So there'll be homework questions where you get instant feedback, and there'll also be longer written response style homework questions. I will provide video solutions to all homeworks. So not only will you get instant feedback, but you'll be able to watch my videos where I talk you through how to answer them and how to get all of the marks. You will get a workbook which you can download in advance of the lesson, so that as I'm teaching, you can actually follow, engage, and make more notes. Kind of like I've been doing on the PowerPoint today, you'll be able to add those notes to your workbook as you go through the lesson. And then after you've completed the homework, you can also watch the video solutions to the homework. You'll join our community of biologists. So they're just there as an additional support. Any advice, support that you need, if you want to share questions or concerns, you can share those with the rest of the students on the course. You also get a 14-day full refund guarantee. So it's totally risk-free. You can sign up to the course, you can start in January, you can come to lesson for 2 weeks, have a go at the homeworks, get the workbooks, and if for some reason it's not right for you, you have got that 14-day money-back guarantee period, so you don't have to be concerned. It's worthwhile just getting your place booked so that you know you're ready to go in January, and there is that no-risk guarantee. We've got some great figures for you here. So 90% of our students recommend my EdSpace. We've got over 1,300 students studying with us in the evenings online, and 82% of those students have, this was last year, came out with a higher grade than what was predicted or what was expected. Basically because they got to go through the content again and do those homeworks and get those video solutions, as well as the workbooks, which basically form the best set of study notes ever. Have a look on Trustpilot if you're still not sure. You'll read all the reviews on Trustpilot about not only our teachers, but about the learning platform itself and the way the courses are structured. So yeah, have a look at the reviews, or get your parents to look at the reviews if they're not sure, and you'll see there are hundreds of 5-star reviews. Put a plus in the chat if you like the sound of this course. Obviously I've spoken to you a little bit about what it involves. I know some of you will be worried about costs and things like that, and you know how expensive private tuition can be. But this course, we are making it accessible to everybody. So this course is £700 for the whole course from January right up until the night before your exams. Okay, in fact, the night before your last A-level biology exam. And we also have a massive discount that we can offer you. That money is not just paying for tuition. Private tutors, gosh, private tutors can charge up to £100 for an hour. And what we're offering you is not just lessons with myself, 4 hours a week of lessons where you can hopefully engage fully with the chat. I mean, I'm really appreciative of all of you that engage and answer my questions, because it lets me know that you're liking it, that you're understanding it. It's really helpful. So live and engaging lessons. You'll also get your workbooks. You'll also get your homeworks, your video solutions. Some of your homework questions, you'll get instant feedback. Other homework questions that are fuller written responses, you'll get talked through with the video solution. You'll get access to the community. The other thing that is great about the course is that all of the lessons are recorded. So you can access those video recordings time and time again. So if you're doing revision in your own time, you can go back and play the lesson on the Krebs cycle and go over it as many times as you want. You will also get the opportunity to have your own personal mentor, which is invaluable. Our mentors really are amazing. We've made sure that they've all got top grades in their A-levels. So they can really help you. The discount we are able to offer you is 44% off, which is really the best discount you're going to get. And this is the last of our free streams, unfortunately. It takes the price down to £393, which works out at just £4.90 per hour. And don't forget, that's not just for the lesson itself, but you're also getting the homework and the study notes and the video solutions. So it's just excellent value for money, because we want you all to have the opportunity to be a part of this and get that A or A star in the summer, just to make sure that you get on to your chosen university degree or your chosen apprenticeship or job, whatever it may be. We know that it's really competitive and you really do need to stand out with those top grades. You can split it into 2 installments as well. So feel free to message after our lesson today and ask about how you do that, if that's something that you think would be useful and help you to be able to afford the course. The discount is valid until midnight on Friday. So you've got just over 48 hours to get your place secured at that 44% off, which means the course will only be £393. Don't forget, it starts in beginning of Jan and it goes all the way up until your last exam in June. The discount code for today is nitrogen. So what you can do, if you go onto the description on YouTube, you can click the link in the description and it will automatically send that text through WhatsApp with the word nitrogen, and that will let my team know that you are here in the free stream and that you are eligible for that massive discount to secure your place. Even if you just want to ask some questions at this stage, feel free to message the team. So click on the link in the description and send that text through with the word nitrogen. A few quick questions before we finish. Don't forget, if you do miss a lesson, all of the lessons are recorded and you can access the lesson on the learning platform as well as the workbook, which we recommend you download before the lesson. But obviously if you miss it, you can still download it afterwards. You can download it, you can print it, whatever works for you. So all of that will be accessible as well as the homeworks on the learning platform. In terms of attention, you can get your own personal mentor. So it's like one-to-one attention. You can ask questions during our lessons and I will, like you've seen tonight, I will answer all of the questions. You can get support with your homeworks from your mentor. You'll also be part of our biology community, so you can ask as many questions in that chat as you want to. So similar really, asking for help. Yeah, ask the community. There'll be students just like you. So you can ask whatever you want in terms of biology or revision or just advice. You can ask your mentor, you can ask me. You can always message the team at My Ed Space. They will respond to any queries if it's a technical query, for example. And I'm really hoping the lessons will be useful. Celeste, that's lovely. Celeste said, thank you, I have loved these sessions. So I'm guessing you have found them useful. What will make our January course even more useful is that you will have access to the workbooks in advance of the lesson. So you will have downloaded them or printed them ideally. You will have that with you, so you will be able to make as many notes as you can during the lesson. And then you'll have the follow-up homeworks and the video solutions. So they will be more useful than these sessions. If for some reason they don't suit you, you have got that 14-day money-back guarantee as well. So you know what to do. Go onto the description, click the link, message the team with the word nitrogen. That will let them know you're interested and they can give you more info or get you booked on. I am hoping that I will see some of you in January because that is our last free stream and our last lesson before Christmas. So have a lovely holiday, whatever you are doing. And I'm hoping that I will see some of you in January where we can start this course properly and go through all of the content and answer all of your revision questions. So thank you so much for joining. I'm going to say goodbye and let you all go and get your tea. I will see you all again soon. Thanks so much for your interaction and all of your questions and answers. I've really appreciated it. Goodbye. Goodbye, everybody. Bye.